हेलो फ्रेंड्स, इस वीडियो लिंक में हम लोग मॉलिक्यूल्स या माइको प्लाज्मा के बारे में देखेंगे। मॉलिक्यूल्स और माइकोप्लाज्मा। मॉलिक्यूल्स और माइकोप्लाज्मा। They were discovered by Knockard and Rocks. Discovered by Knockard and Rocks in 1898. 1898. Mycoplasma are wall-less pleomorphic. Mycoplasma are wall-less pleomorphic gram-negative monerans. Gram-negative monerans. Other name used for them are mycoplasma-like organism. Other name used are mycoplasma-like organism, MLOs, especially the ones parasitic in plants. Parasitic in plants and pleuropneumonia-like organism and pleuropneumonia-like organism, PPLOs. PPLOs because they were first isolated from first of all isolated from cattle suffering from bovine pneumonia. Shape is cockeyed. Shape is cockeyed. Cocobacillus. Helical. Stellate unbranched. Unbranched or branched filament. These monerians can live under. These monerians can live under anaerobic conditions either as saprophyte in the soil, either as saprophytes in the soil and sewage. Are parasite in animal, are parasites in animals, humans and plants. Human and plants. The parasitic form require growth factor. The parasitic forms require growth factor from their host. From their host. Fermentative metabolism is common. Fermentative metabolism is common. Electron transport system is absent. Electron transport system is absent. Cells are without cell walls. Cells are without cell wall. The cell membrane is associated with saccharide. The cell membrane is associated with saccharide including acetyl glucosamine. Including acetyl glucosa mean mesosome is absent. Mesosome is absent. Replicating disc is attached to. Replicating disc is attached to linear. One end of linear. End of but coiled naked DNA. But coiled naked DNA. Two common diseases caused by mycoplasma are two common diseases caused by mycoplasma are which is broom in plant, which is broom, which is broom in plants, and pleuropneumonia in animals. And pneumonia in animals. Now we will describe about Archebacteria or Archaea. Archebacteria or Archaea. Archebacteria. They are most ancient and heterogeneous group of bacteria. Most ancient and heterogeneous group of bacteria that originated from progenotes. Originated from progenotes. Archebacteria evolved when conditions are Archebacteria evolved when conditions on earth were quite hostile. They continue to live under these conditions. They continue to live under these conditions, including deep sea vents. Deep sea are also called as ancient living fossils. Ancient living fossils. Characteristic of Archebacteria. Characteristics of Archebacteria. Cell wall is divided of peptidoglycan. Cell wall is divided of peptidoglycan and cellulose. It is made up of non-cellulosic polysaccharide. Non-cellulosic polysaccharide and protein. instead of bilayer, lipids form a monolayer. Lipids form a monolayer in the cell membrane. In the cell membrane. In cell membrane, the fatty acids are attached to. In cell membrane, the fatty acids are attached to glycerol by ether bond. Glycerol by ether bond instead of ester bond. Instead of ester bond. Fatty acids present in the cell membrane are fatty acids present in the cell membrane are branched chain lipid decrease branched chain lipid decrease membrane fluidity. Membrane fluidity and increased tolerance to extreme of heat. And pH conditions. Now we will describe about groups of Archebacteria. Groups of Archebacteria. Archebacteria have been divided arbitrarily on the basis of their metabolic and ecological properties. Archebacteria are divided on the basis of metabolic and ecological properties. Obligate anaerobic autotroph. Anaerobic autotrophs which get killed in presence of oxygen. killed in the presence of oxygen and produce methane as end product and produce methane as end product. They are called methanogens. They are called methanogens. Facultative are obligate anaerobes. Facultative are obligates anaerobes and aerobes live in hot sulfur spring. Live in hot sulfur spring. They are called thermoacidophiles. They are called thermoacidophiles. Number second, aerobic heterotrophs. Aerobic heterotrophs which live in brine, which live in brine and called halophiles. Called halophiles. Number fourth, we will see about methanogens. Methanogens are obligate anaerobic archebacteria which produce methane as end product. Methane as end product of their body. Metabolism live in swamps, lake sediment, anaerobic sewage digester, anaerobic sewage digester and other similar places where organic matter is undergoing matter is undergoing anaerobic decomposition. Anaerobic decomposition. Number second, halophiles. Halophiles. They live in habitats having high salinity. Live in habitat having high salinity and high light intensity and high light intensity. Halophiles are able to live comfortably. Halophiles are able to live comfortably in this high salt concentration. In this high salt concentrations because their intracellular cell concentration because their intracellular salt concentration is equally high is equally high. The enzyme and ribosomes of halophiles are the enzymes and ribosomes of halophiles are function efficiently only at high intracellular salt concentration. Halophiles possess red carotenoids. Halophiles possess red carotenoids in their cell membrane. They protect the Archebacteria. They protect the Archebacteria from intense harmful radiation. From intense harmful radiations. Now we will discuss about thermoacidophiles. Thermoacidophiles. They are Archebacteria which can tolerate they are Archebacteria can tolerate both high temperature, both high temperature up to 80 degree Celsius and very low pH and very low pH up to 2. Thermoacidophiles have 3 subgroups. Thermoacidophiles have 3 subgroups. Number 1, sulfolobus. Sulfolobus is a chemoheterotroph. Sulfolobus is a chemoheterotrophs which grows in hot sulfur spring. Which grows in hot sulfur spring. Number 2, thermoplasma. Thermoplasma is a wall-less archibacterium. Archibacterium which occur in refuge piles of coal miners. Number 3, thermoproteus. Consist of obligate anaerobes. Aerobes that imply a variety of respiratory substrate. Respiratory substrate. Now we will see economic importance of bacteria. Economic importance of bacteria. Number first, useful activities. Useful activities. Number first, decomposers of organic remains. Decomposers of organic remains. Along with fungi, bacteria are important. Along with fungi, bacteria are important decomposer of organic matter. Decomposers of organic matters. They function as nature's scavengers. Function as nature scavengers. Cleanse the earth of remains. Cleanse the earth of remains of previous generation of organism. Previous generation of organism. Create space and release. Create space and release in organic raw material. In organic raw material for future generation. For future generation. Number second, useful activities of bacteria are sewage disposal. Sewage disposal. Sewage is taken to a special tank. Sewage is taken to a special tanks where organic wastage are decomposed. Where organic wastage are decomposed by bacteria and fungi. By bacteria and fungi. Decomposed sewage is filtered. Decomposed sewage is filtered and the filtrate is used as, and the filtrate is used as manured water for agriculture. Manured water for agriculture. Number third, ruminants and other herbivores. Ruminants and other herbivores. Herbivores take vegetable matter. Herbivores take vegetable matter having cellulose, having cellulose. Cellulose is not digested by animals. Cellulose is not digested by animals. Herbivores instead possess cellulose digesting or fermenting bacteria. Herbivores possessing bacteria. Cattle and other ruminants have cattle and other ruminants have chambered a stomach, a chamber called rumen, a chamber called rumen harbors a number of cellulose. Harbors a number of cellulose metabolizing bacteria. Metabolizing bacteria. Fourth useful activity of bacteria are human flora. Human flora. A large number of bacteria reside over and inside. A large number of bacteria reside on and inside human body. They provide protection against human body. They provide protection against minor infection, minor control putrefaction of undigested food, control putrefaction of undigested food and secrete number of food and secrete number of vitamins. For example, riboflavin, thiamine, biotin, riboflavin, thiamine, biotin, cobalamin, and vitamin K. Cobalamin and vitamin K. Next useful activities of bacteria are pollution control, pollution control. Pseudomonas degrades a variety of pseudomonas degrades a variety of man-made and natural organic pollutants. Man-made and natural pollutants Flavobacterium can decompose Flavobacterium can decompose 2,4-D. Persistent pollutant DDT is similarly decomposed by persistent pollutants. DDT is decomposed by Acetobacter aerogens. Acetobacter aerogens. Now we will see usefulness of bacteria in agriculture. In agriculture. Agriculture. Number first, manure. Farmyard manure and compost are farmyard, manures and compost are prepared from farm refuse, garbage and animal excreta, garbage and animals excreta through decomposers made up bacteria and fungi. Next useful activities: manure and gobar gas. Manure and gobar gas. Animal and human wastage. Animal and human wastage. Water weed and other organic wastage. Water weed and other organic wastage are allowed to undergo an aerobic fermentation. The product of latter are changed to the products of latter are changed to methane by methanogens. The same is collected and used as fuel gas. Simultaneously, the organic wastage are changed to manure. Next utilization of bacteria is in silage and it is preserved fodder. Freshly chopped fodder is placed in silo, sprinkled with salt and molasses to stimulate bacterial activity. Bacteria soften the fodder and enhance its nutritive value. They also produce lactic acid, which preserve the fodder. Next group is It is preserved fodder. Freshly chopped fodder is freshly chopped fodder is placed in silo. Placed in silo. Sprinkled with salt and molasses. Sprinkled with salt and molasses to stimulate bacterial activity. Bacteria soften the fodder. Bacteria soften the fodder and enhance its nutritive value. And enhance its nutritive value. They also produce lactic acid. They also produce lactic acid, which preserves the fodder. Which preserves the fodder. Next useful activities: ammonifying bacteria. Ammonifying bacteria. They are bacteria which degrade protein. Bacteria which degrade protein. Which degrade protein and deaminate the amino acid and deaminate the amino acid into ammonia. Ammonia is trapped in the soil. Ammonia is trapped in the soil as ammonium salt. As ammonium salt. Next, nitrifying bacteria. Nitrifying bacteria. They are of two types. They are of two types. First is nitrite bacteria and second is nitrite bacteria. Nitrite bacteria oxidize ammonium. Nitrite Bacteria oxidize ammonium into nitrite compound. For example, nitrosomonas and nitrococcus. Nitrosomonas and nitrococcus and nitrate bacteria oxidize nitrite. Nitrate bacteria oxidize nitrite to nitrate. Compound. For example, nitrocystis and nitrobacter. Nitrocystis and nitrobacter. Next property is nitrogen fixation. Nitrogen fixation. It is conversion of free or molecular nitrogen. It is conversion of free or molecular nitrogen into compounds of nitrogen. Into compounds of nitrogen. It is carried out by bacteria and cyanobacteria. It is carried out by bacteria and cyanobacteria. Free living nitrogen fixing bacteria. Almost all phototropic bacteria are capable of nitrogen fixation. For example, rhodospirillum and chromatium. Amongst non-phototropic free-living nitrogen-fixing bacteria, Acetobacter, Klebsiella are aerobic, while Clostridium pasturinum is anaerobic. Nitrogen-fixing bacteria are divided into two groups. Nitrogen fixation bacteria are divided into two groups. First one is free living nitrogen fixing bacteria. Free living nitrogen fixing bacteria. For example, rhodospirillum. Rhodospirillum, Acetobacter, Klebsiella, Acetobacter, Klebsiella, Second is symbiotic nitrogen fixing bacteria. Symbiotic nitrogen fixing bacteria. They live in mutually symbiotic nitrogen fixing bacteria. Symbiotic nitrogen fixing bacteria live in mutually beneficial relationship. Live in mutually beneficial relationship. Beneficial relationship with plants. Obtaining food comes shelter. Obtaining food come shelter. And handing over part of and handing over part of fixed nitrogen to the plant. Fixed nitrogen to the plant. They occur as leguminous root nodules. They occur as leguminous root nodules. Rhizobium species. Rhizobium species. Leguminosarum. In the soil, bacteria cannot fix nitrogen. In the soil, bacteria cannot fix nitrogen. After entering the legume root, after entering the legume root, the bacterium induces cell growth. The bacterium induces cell growth, multiplies itself, forms intracellular groups of irregular bacteria surrounded by host membrane, a pink red pigment called a pink red pigment called leg hemoglobin lines the membrane regulates oxygen supply to bacteria. Oxygen supply to the bacteria. Sir, double association. In Sesbania rostrata, nodules occur in roots as well as stems. Root nodules contain rhizobium species, while the stem nodule harbor aerorhizobium. Number third, non-legume root nodules. Frankia forms nitrogen-fixing root nodules in a number of non-leguminous like Casuarina, Alnus, and Rubus. It can also fix nitrogen in the free state. Third is leaf nodules. Nitrogen-fixing species of Xanthomonas and Mycobacterium occur inside the leaves of some plants like Ardacea and Pavetia. Next, useful properties of bacteria are: so friends, we will see other useful properties of bacteria in my next video link. So you wait for my next videos. Thanks.